Yes. Yes. All right. Now I know I need to go in and make my personal Instagram a bit more private. You can see my cats in my crochet. That's fine. I'm happy with that. All right. So I did want to start off today by doing a little bit of review. So I had you watch that video online in order for us to be able to play like a fun game today. So kind of broad overview of that whole video was the different types of ways molecules can move through our membranes, right? We have simple diffusion, you don't need a protein, and this would include like gases, water, hydrophobic molecules, lipids being some of those. It's going to go from an area of high concentration to low concentration, right? It's going with its gradient. We also have facilitated diffusion. Right, so again, we're going with the gradient from high concentration to low concentration, but we need the aid of a transport protein. Right? So this is going to be molecules that can't traverse that hydrophobic interior of the plasma membrane. Right? So it'd be like ions and hydrophilic molecules. Bless you. We also have active transport. So active transport must be something in the air. Active transport, we are going against a gradient, and that's the big thing here, against a gradient. So we're going from an area of a low concentration to an area of a high concentration. And it's not energetically favorable. We need a transport membrane, or a transport protein, and we need the use of energy in the form of ATP. Right? Anything moving against a concentration gradient. Are there any questions about that video I had you watch? Yes, Ava.
This is one. The secondary active transfer.
Mm-hmm.
Wait, never mind. I'll just ask it later.
Sounds good. Secondary active transport can be a little tricky. Any other questions? Yeah.
There's one here is like against the gradient is using energy, but it's like specifically to be able to move another molecule back in. Like it throws out ATP or a molecule that it moves back. Does it only throw it out to be able to get the other molecule back, or throwing it out does it have a specific function in itself?
Cool. So we're talking about active transport. We're moving it against the gradient to build up a concentration gradient in order for secondary active transport to occur. Sometimes it's just to build up that gradient. Sometimes it's just active transport alone. We need to get that molecule either in or out of our cell. Yeah, great question. All right, well, I'm going to have us do just a bit of a review just to make sure if you have any more questions, let me know. But take a few minutes here with each other and discuss the advantages and disadvantages of simple diffusion, facilitated diffusion, and active transport. It was assignment for someone. So it's linked on that PowerPoint, but it's also in the YouTube channel. And the Google Slides. That's okay. She wasn't there. That's okay. Sounds good.
But from the definitions I see, I guess like
It might be a lot of review if you click in the right order.
Oh, that's like the water.
That's our focus. We're not covering that. Yes. So facilitated diffusion is going with the gradient, but you need how you can't just go through the process.
It's going through the osmosis that happens.
Yes. So we will get to that. That's my few slides that I actually have today. Yeah, so you have it up on your computer. That's fine. So against the gradient, so we always want to go from areas of high concentration to low concentration. So there's a lot of that molecule in the and if it's going against it, it's undergoing from a low area to a high. It doesn't like to do that without something important. All right, so let's come back together here. What's the advantage of simple diffusion? No ATP, yeah, that's one of them. What else? Keep going with the gradient. What doesn't it need to use? Doesn't use energy, and it doesn't need, yeah, it doesn't need a transmembrane protein, right? So the cell doesn't have to synthesize a protein specific for that molecule. What are some disadvantages of simple diffusion? Usually just small molecules, like gases, lipids, water can actually do this. Normally they're non-charged. I know water is polar. It's kind of an oddball in that aspect. What about facilitated diffusion? What's an advantage of facilitated diffusion? Yeah, Maddie has it here. Bigger molecules. Anything else? Don't need ATP. What about some disadvantages? You have to make a protein. Yes. Have to make a protein. And so these molecules have to use the channel. Active transport. Right? What are the advantages of active transport? Yeah, it can move against the concentration gradient. Right? Normally these molecules don't like to go against the concentration gradient, but sometimes we have to get those molecules into or out of the cell. Right? What's a downside of active transport? Big one. Uses energy, ATP, yes. All right. We're going to finish off our talking about membrane transport with osmosis today before we end the game. So osmosis is a form of passive transport, so it doesn't need energy to work, right? And when we're talking about osmosis, we're specifically talking about water, right? Water moving across that semi-permeable membrane. Now, water moves according to solute concentration, right? So basically, it kind of wants to equalize the ratio of solute to water molecules. So if you look at this example up here, right, we have our solute molecules on both sides, but we can see this one side has many, many more solute molecules, right? Now we want to kind of bring that ratio to an equilibrium. So water, just the water, is going to move to that other side in order to have that same ratio of solute to water molecules. Right? So we're moving according to the solute concentration. Right? The solutes might not be able to move through that semipermeable membrane, but water can. Right? Water doesn't need a transport membrane. It can move right through the phospholipid bilayer. Right? So we're maintaining this equilibrium. Right? I have another image at the bottom here, just kind of zooming in on that. All right? You see we have our solute, right, and our water molecules are surrounding it. Right? But there's far fewer free water on this side than there are free water molecules on this side. So water is going to move in the direction of the more solutes. Right? So if we're talking about water, you will often see this terminology. Right? So we'll talk about solute concentrations compared to another concentration. Right? So you'd see the words isotonic, hypertonic, and hypotonic. Right? Now we can't say these words unless we're comparing two different things. Right? We can't say that a solution is isotonic. We have to say it's isotonic compared to this other solution. Right? There has to be that comparison. So isotonic would mean that the solute concentration on both sides of a membrane, or maybe the solute concentration in two different beakers, are equal. Right? Equal solute concentrations. Hypertonic, right, that would be this middle picture here, is that the solute concentration is higher on one side of the membrane versus the other side. Right? So in this image here, our solution is hypertonic outside of our cell, right? Because there are more solutes compared to the inside. The last one would be hypotonic, right? The solute concentration is lower on one side of a membrane than the other. And a good example is this last image down here. Outside of the cell, we have fewer molecules than we have inside of the cell. Right? Now we could say in this bottom image that the inside of the cell is hypertonic because there's more solutes compared to the outside of the cell. Right? We always have to compare to another solution or to another side of a membrane. Right? Now this makes a huge difference in organisms. Right? So if we look at, say, these red blood cells here, if we place it in a hypertonic solution, right, it's going to cause the water to leave. Hypertonic solution, there's more solutes on the outside of our cells than the inside. Right? Because there's more solutes on the outside, water is going to move towards that higher solute concentration in order to maintain that equilibrium. Right? This can cause the cells to shrink. Right? On the opposite end here, if we place our red blood cells in a hypotonic solution, so there are fewer solutes on the outside than there are on the inside, water is going to want to move into the cells, right? Because there's more solutes inside of the cells, water is moving towards that higher solute concentration. The result can be that there's so much water that enters into our red blood cells that they burst. Right? The same concepts apply to plant cells. I'm not going to run through this diagram here, but the same would occur. The only reason that plant cells don't burst is because they have this rigid cell wall, and that prevents it from bursting, from swelling too much. So water balance, really, really critical for all organisms, right, including ourselves. So I put an image of some fish here, all right, and so depending on what organism you are, you might be osmoregulating, right, which is the control of your water balance, slightly different. So for example, in a marine fish here, right, there's going to be excess salts around it in the water, right? So water is going to want to leave its body. Compared to this freshwater fish, there's going to be an excess of molecules inside of the fish body compared to the outside because they're in fresh water, not salt water. And so water is going to want to move into the fish. But the fish doesn't need that much water, right? That can make our cells burst, so it has to have a way of regulating its water balance. Right? Same happens in plants. Right? Plants have a whole variety of adaptations to deal with osmoregulation. I did want to point out, right? I said water does not need to transport protein to move, right? It doesn't need it, no, but it can use one. And there is a very specific protein called an aquaporin that is specific for moving water molecules, right? And this protein is essential in a lot of different cells because it makes a rate of water movement faster, right? So if you think about a plant, right, they're going to have a lot of aquaporins in its roots, right? So it can take up water faster from the soil and get it into that plant body. Right? Same with like animals and our kidney cells, we might find a lot of aquaporins because that's where a lot of water balance regulation comes into play. All right, I have this question up here for you. So, if a doctor injected a patient with what was labeled as an isotonic saline solution, but then the patient died, right? An autopsy revealed that several of the patient's red blood cells had burst. Would it be true that the injected solution was really isotonic? Why or why not? And I give you a variety of answers here, and conveniently there is a slide previously to help you out. Give it a couple minutes to think about it. Oh, all right. I gave the answer away. That was supposed to start the timer, and instead it gave you the answer. So I apologize. If you said false, right, you were correct. Right? The solution was hypertonic, meaning they injected a solution that had a lot of solutes in it. So, okay, yes. Hypertonic solution. Hypo? Okay. I gotta think about this too. Let me move back to that slide because maybe I put this one backwards. Right. You're correct. Right? If it had burst, it would be in a hypotonic solution. Oh, all right. So that's an extra added challenge here. I get these mixed up all the time because you have to think about the solution compared to what it's being placed in, and depending on what side of the cell you're talking about, it can have different terminologies. All right, any questions about this? I know I probably thoroughly confused you. All right. The last thing I wanted to talk about is we've talked about movement into cell membranes and out of cell membranes, right? But how do cells communicate with one another? How do we move molecules between cell to cell, right? Well, these cells do have these intercellular channels that allow for the direct movement of molecules between adjacent cells. So these are cells that are snug up against each other. They are touching, right? They have channels in them, right? So it's called a gap junction in animal cells, where they can directly move one solute into the opposite cell, right? It doesn't have to go outside of the membrane and then back inside of another membrane, right? Those two membranes, they're snug against each other. There is a channel connecting both of those phospholipid bilayers together. It moves one cell to another. It doesn't have to go outside of the cell at all, right? The same thing we find in plant cells, what we call plasmodesmata, right? It's still a junction, a plasma membrane-lined junction between our two adjacent cells. Now, sometimes we do have to take in molecules from the outside environment, right? Or excrete it to the outside environment. And we briefly touched on this when we talked about, say, the Golgi apparatus, right? And so if we need to transport large materials into or out of the cell, cells often employ this process called exocytosis, right? It's where material is nicely packaged. You can see there's the Golgi apparatus there because the Golgi apparatus often is the organelle that packages a lot of these larger substances. It's packaged into this membrane-lined vesicle, right? It moves throughout the cell. The vesicle will join with the plasma membrane and then release its contents. right? Now the opposite is true if a cell needs to take something in, right? That would be endocytosis. And this is where the plasma membrane folds inward and kind of encapsulates something. There's different types of ways of doing this, right? I'm just going to show you an example of one of them, which is this receptor-mediated endocytosis, right? So if it's a receptor-mediated, that means we have receptors embedded within our plasma membrane here, right? They are sensing the molecule that needs to be transported. Once that molecule attaches to the receptors, the plasma membrane folds inwards to create this vesicle and then transports our cargo to where it needs to go, right? Oftentimes this could be a lysosome. It's attaching to a lysosome in order to digest maybe food particles. Yes? Which protein? The coat protein. Yes, I believe the coat protein helps it regulate to where it needs to go. I need to look at it. Yeah. Good question. Any other questions? All right. I did just want to give you an update of homework here before we go outside to play a game. Content check number 2 is due on Monday. Right? I will do that last question in class. I will say this last question is going to involve drawing. Right? So make sure you at least have a sheet of paper ready to draw. You can turn that into me, and you can turn the rest of digitally, whatever works for you. Otherwise, the scientific literacy assignment is due Wednesday, and then you have your exam next Friday. Yes? It is during class. Yep. So I'll be here. You'll have 50 minutes to do it. Like on paper? Is that the type of format? Yeah. Paper format. I want to say, I haven't made it yet. Probably been about 4 or 5 pages long. I know that sounds like a long test, but that gives you room to answer short questions and for me to answer maybe some images for you to talk about. I so far have not had too much difficulty with students not finishing on time. Right? I tend to be able to time these tests okay. If I find that nobody has finished in 50 minutes, I will take that into account when grading, okay? So don't worry about late. All right, it's a beautiful day, so we're gonna go outside. I wanna briefly explain the game to you before we go outside, just so you have a visual to help you through here. So I'm going to give you all a molecule, okay? I will pass those out when we get out there. You do not know what your molecule is. Right? You have to ask your classmates questions about your molecule in order to figure out its properties. Right? Now, I don't expect you to guess the molecule, right? But I would expect you, by the end of your questioning, to figure out, does it move by simple diffusion, facilitated diffusion, or active transport? Right? Once you have figured it out, right, you will go next to the group that represents each of those 3 types of transport, and then you can open it up and see if you agree with your assessment or not. Okay? We'll play this once for sure, a second time if we have time, or there's people who want to play. And the answers for all of these molecules will become available on Moodle at 1:30. So you can go back and use that as a study tool. Yes? No. But I would expect you to know, like, thinking about, is it polar, is it an ion, is it a gas, is it small, is it large? Think about those properties of a molecule and how they would apply to different transport types. But I would not expect you to know that this particular molecule moves in this particular way. All right. Let's see. I was thinking of a good place to go outside. I don't know if we can actually go on the grass out there because they just planted it. What about, I think there's a spot on the end of the building here. Does that seem reasonable? You can walk out and around. All right. Give me a couple minutes for me to get my computer disconnected.